Okay, welcome to part 2 of 3.3 Meiosis for IB Biology. So last time we left off reviewing mitosis, right? This was mitosis. This is where we're starting with 2n, or diploid, number of chromosomes. Paying attention to the number of chromosomes is very important in these sections. Okay, and then we still end up with diploid daughter cells. They are exact copies of each other. But that is not the purpose of mitosis. Or sorry, that is not the purpose of meiosis. So with meiosis, we are starting with parent cells, right? One parent cell. And we're going to start with these replicated chromosomes. Again, these are the X-shaped replicated chromosomes. Okay. Okay, and I was trying to draw those over here as well, so you can think of them as also being X-shaped over here in mitosis. So we've got the X-shaped chromosomes. We call these, what do we call them when we have the pairs of chromosomes? Remember I always said there were 23 pairs of chromosomes in humans, right? What do we call those pairs? Chromosomes. We call them homologous chromosomes, right? So these are the 23 pairs of homologous chromosomes. So I'm only drawing 2 pairs here because I don't feel like drawing all 24 of the pairs. But that is what this is supposed to represent. 23 pairs of homologous chromosomes. Okay, they are replicated right there as well. We have one replicated chromosome here, one replicated chromosome there. And if they are lining up, which phase of mitosis or meiosis would that be? If you said metaphase, you would be correct. They're lining up in the middle, metaphase. Okay, so another way of saying these 23 pairs of chromosomes or 46 chromosomes, and remember they are all replicated as well, is you can say that they are diploid, right? Or 2n, right? Okay, the next step is it goes through one round of meiosis, right? So we call this meiosis 1. At the end of meiosis 1, you do end up with 2 cells that would contain what? They contain these replicated chromosomes still. Okay? But you'll notice that how many chromosomes are up here and how many chromosomes are here, how many chromosomes are here. You'll notice that right here we have 4 chromosomes. They're 4 replicated chromosomes, of course, because they're in that X shape. Over here we only have 2 chromosomes. Okay? 2 chromosomes. And over here, we also have 2 chromosomes. And they're replicated still. But that shows you that the number of chromosomes is what? They're halved, right? So what do we call it when you have half the number of chromosomes? Haploid, half, or 1n. Right? Okay. So that's only after metaphase 1. Right? So let's make sure that we know. Sorry, not meiosis 1. Right? That's what's happening right there. Okay? And then we're going to now do meiosis 2. And so the results of meiosis 2 are, it's essentially exactly what ends up happening in mitosis. Meiosis 2 looks basically exactly the same as mitosis. So we end up with some daughter cells. Right? We end up with how many daughter cells though? We started with 1, and we're ending up with 4, right? And these 4, are they identical? Are they exact copies like over here in mitosis? They're not, right? So we separate these replicated chromosomes into single chromosomes. And so now we have how many chromosomes in each of these cells. Don't forget, when they're replicated and the sister chromatids are connected to each other, it's 1 chromosome. But when we pull them apart, they do become 2 separate chromosomes. So now we've got 2 chromosomes, 2 chromosomes, 2 chromosomes, 2 chromosomes. We still have 2 chromosomes here after we did mitosis 2. So you end up still with 2 chromosomes, just like you had over there, right? This has 2 chromosomes, right? This has 2 chromosomes, okay? 2 chromosomes, and 2 chromosomes. Meaning, is it haploid or diploid? It's still also haploid, right? 1n. Each of these cells is haploid or 1n. Okay. Don't know why it keeps doing that. Okay. Okay, so now let's go back to some of these statements and see which ones we've ticked off, which ones have we covered. 1 diploid nucleus divides by meiosis to produce 4 haploid nuclei. I would say that we've covered that one, right? Okay, how about this one? The halving of chromosome number allows sexual life cycle with fusion of gametes. We actually haven't talked about that, so let's go back to our drawing. These are now considered gametes, okay? So in males, those are sperm. In females, they're eggs, right? And so you end up with 4 gametes with half the genetic information, haploid, than the original parent cell. Why do we need half the genetic information of the original cell? If you said so that the gametes can fuse, so that sperm and egg can fuse to complete the life cycle of an organism by fusing half the genetic information from the mother, half the genetic information from the father, you would be correct. That's what this statement's all about. How about over here? DNA is replicated before meiosis so that all chromosomes consist of 2 sister chromatids. Right? Remember we talked about how in order to get that replicated chromosome, this replicated, this one, right? Because remember, the X shape is a replicated chromosome. In order to get that, you actually have to go through DNA replication, which we have learned about, right? And so that's what that statement's all about. It's just saying DNA does have to be replicated before meiosis. It also has to be replicated before mitosis. Okay? And then early stages of meiosis involve pairing of homologous chromosomes and crossing over, followed by condensation. Okay, we have not talked about that. I'm going to get to that soon. Orientation of pairs of homologous chromosomes prior to separation is random. Okay, this is something that we could look at right here. This is essentially saying that when these do line up, when the homologous chromosomes line up right here, if this was metaphase, for example, it's random, okay? So you could have this chromosome could be up here, this chromosome, right, pair, could be down here. And they're going to go through something called crossing over, which we're going to talk about later, that increases genetic diversity and genetic variation. Okay, and so it's just talking about that orientation and the lining up of the pairs of homologous chromosomes, which is random. Okay, the separation of pairs of homologous chromosomes prior to separation of pairs of homologous chromosomes in the first division of meiosis halves the chromosome number. We did talk about that, right? In detail. Crossing over and random orientation promotes genetic variation. Okay, we're going to get there, to crossing over and random orientation. Fusion of gametes from different parents promotes genetic variation. Okay, essentially what this is talking about, and you'll see mention of genetic variation very often because it's an important concept. Genetic variation is important because why? Because for evolution and for adaptation to occur and for natural selection to occur, right, we need genetic variation. So this is just saying that because different parents are contributing half of their genetic info, random half of their genetic info. For example, your mother contributed a random half of her genetic info. Your father contributed a random half of his. And this is promoting genetic variation because of these two random halves of genetic info that are coming together to create a new organism. Okay, non-disjunction causes Down syndrome. We will get there. Studies showing age of parents influences chances of non-disjunction. We'll get there. Methods to obtain cells. We actually have talked about this. We talked about this in 3.1, right? With chorionic villus sampling and amniocentesis, okay, and associated risks. Drawing diagrams to show the stages of meiosis, resulting formation of the 4 haploid cells. We somewhat drew this. We didn't draw the stages in detail. We'll get there.